Okay. Wait, I don't have I don't have anything to hold. Okay. Hi guys. Hi. Welcome to my Boltzmann session. Wait, where's the computer? Yeah, I'm asking a very oops. Right. Thoda. Yeah. Okay. There we go. Yeah. So yeah, thank you for coming, guys. Am I audible, or should I like move my mic? Yeah, I hope this is better. Okay. So yeah, the aim of my talk is the title of my talk is very vague. So I'll explain a little bit. My intention is to connect astronomy and biology to like explain a little bit about the origin of life. We are here as complex organisms. There's a lot of theories on like how life really evolved, and like just instead of saying how, like even like all the hypotheses we put are just hypotheses. We don't have definite answers. It's not like experiments we do in lab to find cures for things, and we get to know because there's multiple ways how things could have ended up to how we are now. Like Darwinian evolution was one of the theories until it was repeatedly proven multiple of times in different scenarios that we now like believe it as a strong concept. But like we've never really found something very strong, but there's a lot of research that's done on origin of life, and I'll be showing like and like in just without stopping at how, we'll go a little layer deeper and ask why. See if physics science has the answer to it. So yeah, why are you alive? Like take a moment to think about that question. No existential crisis before end sims like Nanda was expecting, but yeah, please don't get any crisis. Let's not see it philosophically. Let's see it physically and chemically. Think you guys are experts. Assume yourself to be expert in certain fields. What if you're a biologist and you're a physicist? Okay. If you are asked why are you alive, what would you say? Why would you say is the meaning of like purpose of life evolving?
For a biologist, like reproduce your organism.
Yes. Okay, that's a good approach. Reproduce and like give the next offspring. An evolutionary biologist would say we are alive to pass on our traits to the next generation. A physicist would say to okay, that would be a spoiler for my talk. Physicist comes later on, guys. And a philosopher would ask even more complex questions about why are we alive. But I'll not answer this yet. We'll get to this later. But before that, let's zoom out. Let's zoom out and look. Have you guys seen this image before? So this is one of the earliest images taken by the James Webb Telescope. And I'm sure this image has like tons and tons of galaxies and stars. You can spot a couple of galaxies. And like to imagine, like I think when we were kids, like we know something's big or heavy, we don't know how, like we can't give estimates. Like we can't know the difference between 10 kgs and 15 kgs, but over time we experience lifting things and relating them to weights, and then get a scale of how heavy or how big things are. But I don't think anything would ever, at least for me, nothing ever gave me the realization of the scale of the universe. We say it has billions of stars, like sun is 150 million kilometers away, but where's the scale of the number? It is huge. But so just to try to give some scale with numbers, because we like to work with numbers, I've put a number on the slide. It's 77 followed by 22 zeros. Do you know like what that number is called? Like 7 septillion. Number is fancy but still doesn't give me scale. But yeah, here on the screen it's like very big. So these many stars exist, and yeah, like I think we can see a couple of them here in our image. So let's ask, among these many stars, I'm sure, like for sure, our star has life. Our star, I mean, like we have Earth. Our star has Earth, and Earth has life. I hope somewhere else there is life, but like like, if it happened here, why is there not a chance that it could have happened like elsewhere too? But then like there are other factors. We are very far, and like the conditions are different. So let's go over seeing like what are the conditions required, and what do you think if even if the conditions are met and there are civilizations outside, can we ever reach them? Like these are all big questions that never got answer to. So someone in the 1950s asked this question: where is everybody? In a casual conversation at dinner table in Los Alamos. You guys know Los Alamos? What happened in Los Alamos? Yeah, the Trinity test, exactly. So this was one of the people involved there. He literally like derived half of particle physics, I think. It was Enrico Fermi. So Fermi asked, where is everybody? And like probably not at the dinner. I don't know if he asked it in the context of where is everybody at the dinner table or where is everybody like in the grand scale of the universe. But what we know here, yes, he's asked about the universe, and this is the origin of the Fermi Paradox. Well, nobody had an answer then. Even now we don't have an answer, but we have some hypothesis, and let's see about that. So we've all, I think, like most of the astronomy enthusiasts would have seen a certain equation about finding the probability of us contacting alien life, and that is this. So that's the Drake equation. It's the probability of communicating other like life within our galaxy to check if it exists. Okay, I think I got the wrong equation, because it says the amount of bullshit you're willing to buy from Drake. We are not believing Drake, guys, because you might assume it says an equation, but like equations what we work with involve numbers and values, but this equation wasn't really intended to talk about numbers. So apparently this person, Frank Drake, he was one of the pioneers in starting the SETI program. SETI stands for Search for Extraterrestrial Intelligence. So in their first annual conference, he wanted to set some agenda to what do you have to look for if you want to find alien extraterrestrial life. So that was the reason he found the Drake equation, to explain the factors which would be involved in us finding extraterrestrial intelligence within our galaxy. So don't look at it at what are the values each of these factors could be, but think of it further as these are what went right or need to go right in order for life to evolve. And then like, even after life evolves, you have to be advanced enough to send messages and receive messages from each other across like intra-galactic timescales. So to know if they are advanced enough, you would have heard the concept of a great filter. A great filter is the idea where there is civilization grow around, but they are not able to develop to a point where they are able to send signals or like communicate with other life, because either they are not able to get their technology as advanced, or they are not surviving till then, or they are not intelligent enough. A lot of factors can exist, but I feel the probability of a civilization being able to reach, if it exists elsewhere, is if it turns into an advanced civilization. You guys would have heard of the Kardashev scale. So that scale is to define how much of energy civilization is utilizing from its parent star. And a type 3 civilization is something which could utilize every bit of energy that is coming from its sun. So you would have heard of Dyson spheres, where they are building an entire sphere to harness the entire energy of the sun. What number do you think we are in the Kardashev scale? Do you think we are between 1 and 2 or less than 1? 0. We are close to 0.8 apparently. We are able to harness a decent extent of the sunlight coming from Earth. We would be a type 1 civilization if we are able to collect all the energy that is coming from the sun on Earth. That would make us a type 1 civilization. So first I think we should be able to get there in order to be like fully energy independent, and then try to communicate around. Okay, so I am saying that in order to understand if there's really life outside, like it's enough of seeing outside, because there's a lot happening. Everything's very far. Time scales are very skewed, because by the time if Toshit is in Betelgeuse and he sends me a signal, by the time I see the signal, I don't think Toshit will be in Betelgeuse. So let's look inward. So I like to make a telescope to microscope analogy here. Let's put our telescopes aside. Let's pick up some microscopes. Let's look within to understand what is really needed for life to form. And maybe that would help us understand what do we really need to look for. Do we really need to look for signals, or we need to look for something more, like first the building blocks of life. So let's get inward, guys. So let's zoom in now. We had zoomed out, looked at the universe, looked at a galaxy. Now let's zoom in. And there's this one molecule that is so small that even a human hair is so big. The molecule is so small you can't really see it using a microscope. And because if a light microscope can't see it, then you can't really even define a color to it. So what I'm talking about is RNA. So these molecules are subcellular. If someone really shows you an image, a light microscope image, and says, guys, this is DNA or RNA, don't believe them, because I'm sure it's probably chromosomes, the entire chromosome, the DNA wrapped around proteins and compressed. You can see that. You can see there are strains that bind to DNA, and then you can visualize them. You guys have sure done onion peel experiments, and you would have seen nucleuses. And sometimes if you stain the nucleus enough and you let cells divide, you can see mitosis happening too. So you can see the chromosomes. But that's as far as the resolution we can see of DNA. Well, you can't see individual nucleotides and stuff with regular microscope. So we have other techniques such as nuclear magnetic resonance, NMR, and electron microscopy. So just I'm telling all this because to give you a scale of we are going deep in where it's into biological processes, and let's try to understand them. That's the problem. Let's cover the problem of life. I'm saying that life shouldn't exist. But still here we are. Not like it did not happen because I say it shouldn't exist. I'm saying it shouldn't exist because it's such an improbable event, and let's try to understand why. Okay, just have a look at this video. It explains the central dogma of biology. People would have done basic their life sciences courses here, and would have who did life sciences. Oh wow, a lot of you have done life sciences. Okay, so you know a little bit of biology. For people who haven't, watch this video, and then I'll tell you a little bit about what the central dogma is. Okay. What do you think that is? There's a long strand. No, it's not protein. Okay, the long strand is DNA. And what is coming and binding to it is protein. And why is this happening? So what is, so I'm pausing the video guys, I'll explain you. I think because I'm streaming it from YouTube, it's doing that. Okay, so that okay, that is the DNA strands, and what went and attached to the DNA is something known as RNA polymerase. So this RNA polymerase is reading DNA, and with whatever you guys would have heard, like DNA is made up of ATGC molecules, and it'll read that, and whatever if it is an A, it will then the RNA polymerase will place an U in the place of A, and for T there's no for a T it will put an A in the RNA. So these are complementary pairs, and this RNA strand that is formed will be the messenger, and this RNA strand will leave the cell. Let's try going there. Not able to forward the video, guys. Okay, I'm not patient at all. Finally. So yeah, you can see the polymerase binding to the DNA, and then it extends. And along the way, the yellow thing that is getting spewed out is RNA, and this is real time. Think how fast it's actually happening in your body right now. That process is literally happening in your body, and it's happening that fast, and it's spewing out the RNA. And not all RNA that is made is usable. There are things called introns and exons, where the mRNA which encodes for protein and then which doesn't. The green part is an exon. I'm sorry guys, I forgot my bio. It will get cut off, and it will merge the yellow parts together, and the yellow part of the RNA will now leave the cell. You can see it's leaving it leaves the nucleus. It's leaving the nucleus, and it goes to the ribosomes. And the ribosome, as we know, will help in reading the mRNA that exited the nucleus, and would produce proteins. Okay, I have finally explained central dogma to non-biologists in a very bad sense. If you have doubts, please come back to me. But yeah, I think let's continue this. Okay, so that was the central dogma. We have DNA inside the nucleus, and not all DNA is used. It's the archive, it's your library, and whenever necessary, if you want to make a certain protein, the mRNA gets transcripted. We have the mRNA now leaving the nucleus, and the chef is the ribosome who cooks the protein and cooks the protein. Reminded me of protein mess, I didn't eat yet. Who synthesizes the protein, and now the protein will do certain functions. If it's melanin, it will give you pigment in your hair, or if it is insulin molecule. No, I'll not get there. What other proteins are there? I'll just leave the example at melanin. It gives you pigment to your hair. So this is the idea. But it is not central dogma. Like all rules must be broken. Central dogma also has its flaws. Some some RNA long back organism found that it can hijack this mechanism and make it not happen by doing something called reverse transcription. It can use RNAs to synthesize DNA. So that's what something called retroviruses do. So these retroviruses have what is an RNA template which has instructions to make more of RNA template, and also something known as a reverse transcriptase enzyme. It also has instructions to make the reverse transcriptase enzyme. When a virus attaches to a cell and releases this, it uses our own cellular machinery to cut it, to integrate this into our mRNA, so that when it is being synthesized, it will synthesize the reverse transcriptase, which will then make DNA which the retrovirus wants to spread, so that it can make more copies of itself. Finally, retrovirus is also using our machinery to make more copies of itself. That's why you would have studied in school that viruses are neither dead nor living. Viruses contain what is necessary for life, but they're not really alive because they don't have the entire cellular machinery. And something else also does not follow the central dogma: ribozymes. This is where it gets interesting. So these ribozymes, let's get to ribozymes in a moment. We talk about evolution, but evolution in the sense Darwinian evolution. We know how we humans evolved from less intelligent organisms, and before that different species altogether, and the whole survival out of the fittest thing happened, where so that it's necessary because our environment is constantly changing. So each and every generation will have certain changes which lead to it surviving further. So some people survived, some didn't, and somehow till now we are all alive. But then how did this whole evolution thing happen before even a cell formed, before even something that could self-replicate come up? How could it have undergone Darwinian evolution? So that is the question. So if we have a cell, we are to build the ribosome. Ribosome is also just made of proteins and RNA. But to make the protein, you need ribosomes. So it's a chicken and egg problem. We don't know what came first. So there is a hypothesis known as the RNA world hypothesis. Okay. Before getting to RNA world hypothesis, a mathematician saw that DNA should be the information carrying agent in a cell. He predicted it before even DNA was discovered in a cell, that DNA contains information. There's something should contain the information. This is the mathematician. Does anyone know him? And this is what he proposed. He is someone who proposed the conditions for a self-replicating machine. So it's John von Neumann. It's Neumann. I think he's had contributions in game theory, mathematics, and a lot of basics of robotics. So when he said that a machine has to self-replicate, there are three conditions. The instructions for building the machine is to be needed. So the first point, and then there should be a mechanism to read that information, and then third, there's a mechanism to build from the information that you're reading. And we can see that before even DNA was discovered, Neumann had framed this, and that is exactly what is happening inside our cells too. Our cells have optimized to be a self-replicating machine. So let's, like I said before, ribozymes are the self-replicating machine. And the RNA world hypothesis states that before the instructions were split into three separate machineries, DNA, RNA and protein, to replicate an entire cell. It was all done by the same molecule, a ribozyme. Ribozyme is basically an RNA which also acts as an enzyme. Think you have an RNA strand, and that could help itself in attaching if it finds new nucleotides, help it attaching himself and make more copies of itself. So that would have been the starting point of a self-replicating machine. And in 2024, there was a research done to discover the way they discovered that RNA catalyzed Darwinian evolution was proven, where they did use these ribozymes and let it undergo changes and mutation. And they are saying that just like how our Darwinian evolution of living organism happens, these ribozymes also ended up evolving in a similar manner, where whichever enzymes could help replicate on their own without loss of information, they were sustaining, and they are what formed the building blocks of the current life. So this is a small meme about the RNA world hypothesis. RNA had its time, but now we are in the central dogma world. But are we really in the central dogma world, or do we still see remnants of the RNA world hypothesis? For that, let's look at the ribosome itself, which produces proteins. Ribosome, we can say, is a protein, but still it is an RNA machine. Ribosomes have two components. Let me zoom in here for you. So you can see the ribosomes have the yellow section and the blue section. These are all proteins. These are structural components which the ribosome is using. It's using to stay in shape. But then the functional component of the ribosome is still the RNA. The green and white part you see here, and the magenta part you see here, are all RNA. So a ribosome still has RNA. So finally, despite it looking complex, the core principle of ribosome function is RNA. So I added a small slide to talk about RNA because I work with RNA in the lab. I work on understanding how to stabilize RNA in room temperatures, or to understand how to prevent degrading them. So even DNA. So RNA is a very fragile molecule. Like our sweat, our breath, all contain what is known as RNase, which is an enzyme which breaks down RNA. Or even temperature fluctuations from outside, or stresses, can lead to RNA degrading. So such a sensitive molecule, how we handle them is wearing gloves, wearing masks, and I use a chemical called RNA zap, which makes sure all the RNAs in my hand is in my gloves are gone. And we do something called autoclaving in the lab, which is a sterilization method where you put all your stuff which you're going to be working with in a pressure cooker and take it to 121 degrees for 20 minutes, and that temperature and pressure will break down all the proteins, and your equipment is sterile. But this RNase enzyme apparently is very stable, and even if at that high temperature it unfolds, if it denatures, it will renature and be functional. So working with RNA is really difficult. But a molecule that is so sensitive, how did it really form as the basis? As we just saw that ribozymes, which is basically RNA, is the building block of all life. How did it really form in the beginning? It is thought that it was formed in somewhere where water is there, and it had the right conditions for these molecules to attach together, or we don't really know how it happened. Okay, going to the next chapter. We all know about the second law of thermodynamics. What is the second law of thermodynamics, Ganesh? Okay, anyone else? I wanted to reference Aditya Mohapatra. If AM would have been here, he'd be so proud. There's a band called Muse. So they have some song called explaining about the second law of thermodynamics. And the whole law states that the entropy of the universe is always existing. So that's the law, guys. So everything in the world, if it really leads to a disorder, leads towards the entropy increasing, why did life really happen? Isn't life like very ordered? We are structured, we have mechanisms, we have information. Okay, Kirtana and Ayana are saying they are not structured.
No, I think structure doesn't necessarily mean that it doesn't increase the.
Yes. Ayan? Yeah.
Aren't humans creating more and more disorder? I thought
That's the whole point. I think we are putting enough nukes to balance out the order that we are all built upon. So yeah, that's the basis of the topic that I'm going to come now. So the twist now, we saw what's the problem. Life is very improbable to have evolved, but my twist is physics is like it's not like life is not really disobeying the laws of physics. It is what is running it. So I'm showing an image of a hurricane, and I would like to ask a question. What would you say are the characteristics of being alive? Like life, if something is alive, how would you know it's alive? Sorry? Response to stimuli. Okay. Anything else? Huh? Self-replication. Okay. Sex. Not necessarily. Only sexual, like asexual reproduction does happen. So why I put a hurricane here is to like draw parallels, which I think I'm gonna do very badly, with like why wouldn't hurricane not be alive but cells are alive? Let's see if I can sell that to you guys. Huh? Can't reproduce. Hey, that's a good idea. But I thought a hurricane could split into two tiny hurricanes and I could call it mitosis of hurricanes. But yeah, but the idea is hurricanes are also causing disorder. We are also causing disorder. Okay, let's see what's different. So, like Ayan and Ketana said, we are like creating order within, but then in the end there is a lot of disorder created outside. But what is really running it? What is really pushing evolution to make us, like why is life the answer to increased disorder? Let's see that. So Schrödinger in 1944, like someone he's a quantum physics guy, but bro wrote a book on life. That's like me trying to study quantum mechanics and I don't understand anything about it. So I went to the abstract of the book, couldn't read it fully, but his ideas are really good. He coined the term negtropy, negative entropy. So his whole idea was we feed on the negative entropy of our own system, but then all the other actions that we are doing is increasing the entropy of the entire universe. So we are, despite like reducing our internal entropy, we are able to increase the entropy of the universe. And he also in this book mentioned something called the aperiodic crystal. So before, like it's 1944, it is still long before DNA was discovered to be the fundamental unit of like containing information about life. But he said that something like a sugar crystal or a salt crystal that has multiple repeats, that has no information. But then life has to have something which is an aperiodic crystal, and this non-repeating pattern is where the information is. This comes around the concept of Shannon entropy. I don't know much on how to explain Shannon entropy, but me and a friend, we were having a discussion about Shannon entropy, and he gave a very beautiful analogy. Let's say you walk into my hostel room. It's so messy. Things are randomly here and there, not at all organized. So my friend, if he is new to the room, he will see the room as disorder because he has no information about which is where and what is placed where in the room. But then I myself have information about where and what the room has. So just because he sees the room as chaotic, I won't say the room as chaotic. Like there is certain order, even though it is not periodic. So that is similar to how we should see DNA. For us, the molecules, the nucleotides seem gibberish, but there is some information present in that which life can use to self-replicate. So this person, Jeremy England, he also explained something about life, that why physics is really optimized towards like organized life, because we seem to be better at absorbing and dissipating this energy, and self-replication is the best way to have this energy kept on dissipating. So life might not really be an accident, and every cellular process is governed by physics for sure. Nothing disobeys physics, and nothing can escape the second law. So I think it is supposed to have, it was like bound to happen that life does turn out to evolve if we want entropy to keep increasing. So I wanted to put a video here. I could not put that. Good. Now I can go to YouTube again. Okay. So this is a video I wanted to show of molecular dynamics simulations. These are stuff I do in lab. What you see here is a box. All these red dots are water molecules, and inside you can see something colored. So inside the box we place a protein, and you'll ask like, how are you doing it all computationally? So what we are really doing is we are associating probably weight, charge, lot of parameters. There are packages which have all these parameters associated with atoms. So we feed in, if I want a protein, there's a database which has all the information for a protein, which atom and which coordinate it is. And if I load it into the software, I'll get something like this. So, and these simulations do basic Newtonian mechanics only. Like all F is equal to ma. So what happens is, our time steps of my simulations will be in femtoseconds. So it would shift. It will calculate the interaction between two atoms or a group of atoms around, and then see how much force is there, and after a femtosecond shift it a little bit and does the calculation and does it all over again for like thousands of steps. I run like a 50,000 step simulation sometimes, which might seem less but uses a lot of compute, and which might be the reason your GPU prices are going up too, because we need them for simulations. And so the idea, what I'm trying to say is, like in MD simulations, what is happening is Newtonian mechanics only. So, and you can see these molecules jiggling. The ligand is like sitting in the pocket of the protein. So this might seem very like chemical, like you're looking at the chemical realm, like there's a protein, some interactions happening, what's very fancy. But all this happening again and again and again is the real reason why biological systems are running on the outer scale. So like I showed this to show the fact that, like, even though biology seems very complicated and we can't really like compare them to physical behavior, like in the scale of protein interactions, but inside, protein folding or protein ligand binding are all completely governed by physics. And I want to say something about DNA also. So, just like I was telling about Shannon entropy, the information and the sequence, the information is encoded in the aperiodic sequence, and like that is why we, with all our hard disks and bits, are able to store some terabytes of data in a 250-gram hard disk. But DNA optimized it so well that 1 gram of DNA could store like petabytes of data. So it just has its own language. And so as we saw the aperiodic crystal concept, the information is learned. It's earned. It's earned by evolution. Like it did every time the molecules that do not survive get left over. The only ones which do have certain new information. And just like that, over like billions of years of organic life on Earth, the DNA accumulated a lot of information on how to be good at self-replicating. And that is all the information that is contained in the DNA. So, where did this information come from? Like the atoms, as we were discussing, the arrangements of the atoms are the only thing that is changing. The atoms are the same. If you rearrange them or they will put apart, they have no significance. So that we don't know. We just know it's getting better and better when we are evolving. But then how it started, like how the first ribozyme really came up, we don't have an answer to that yet. Okay, zooming out one last time, let's go back to the cosmos. We have Earth and we now know what is really needed for life to get started. We saw that you need amino acids. I did not add slides like the Miller-Urey experiment because I think those are established facts. I will briefly graze through that if you want. So long ago, Earth had carbon dioxide, nitrogen, oxygen, carbon, all separate, but then a lot of lightning and thunder like energized these atoms, rearranged them, and that's how the first organic molecules came out. And these organic molecules somehow rearranged themselves to give life. So in Earth, they found something called hydrothermal vents. These are vents under sea. There's no sunlight, there's no energy, but then they still found life there. So how would life have come out there? So, and there is some parallels that I like to draw with the microbial life they found there and microbial life, like our own cells. Our cells have something that's widely known in the memes as the powerhouse of the cell. What is it? Mitochondria, yeah. Do you guys know that this mitochondria was years ago a cell of its own? So, like long back, prokaryotic cells, some cell engulfed this mitochondria, trying to befriend it, saying, you give me what I need, ATP, and I will give you in return. So that symbiotic relationship led to mitochondria being engulfed by a cell, and that is all our common ancestor to every life on Earth. So why is it similar is mitochondria generates energy by by proton gradients. There's a concentration gradient and it uses that to generate energy which it will use for sustaining. The microbial life they found in these hydrothermal vents were also very similar. Because of the extreme temperature difference and presence of hydrogen gas, there were proton gradients which these life used to stay alive. So if life could really form without any sunlight deep in the sea, this is a similar environment that we found far from Earth but within our own solar system. Does anyone know what picture this is? Enceladus, yeah. So this is one of Saturn's moons, Enceladus. They found that there's a lot of plumes flying away from Enceladus, which is a result of hydrothermal vents from the undersea. It's an icy moon of Saturn, and this moon is also hypothesized to have icy hydrothermal vents, and they are spewing out into the space. And there's also a mission to fly by Enceladus to go touch and see the plumes, to find what is the real composition of this. And they found that this has water and organic molecules that could build what is life that is on Earth. So it had liquid water, it had organic molecules, also had hydrogen. So it had everything that we see what is similar to the hydrothermal wells under Earth, under the sea. Which organic molecules? Not very sure. I don't think it's similar to the amino acids that we have here. That much I know, but I should check further. So similar to Enceladus, there's also Europa. NASA launched Europa Clipper. They are also suggesting that there's hydrothermal activity in Europa. So if they do flybys, we'll get more information on knowing what organic molecules Europa has. So there's one other proposal which is way off the idea which we are discussing right now. It's called panspermia. So we are saying life really formed like on Earth or in Enceladus or Europa, but then there's a hypothesis that life never really formed here at all. It formed somewhere else and it hitchhiked through meteors. So that is panspermia, where meteorites are the passengers which brought in organic life on Earth, and then all the evolution which we discussed happened. This seems more probable because we are very unsure about how the organic life began in here, in complex forms. But there's not much research you can do to explain this. They found different amino acids. See, even other than the 20 amino acids that we humans need, the Earth also has around 200 amino acids which are not in current, which not used by life. So are these amino acids really that significant or not? Can't really be tested. But this is still a hypothesis that life could have really come from somewhere else before it is really life. It was just a group of molecules. So every living thing uses the same genetic code. So that is something very interesting. The information to make each amino acid, this was established long before any evolution happened. All the life you see in here is from DNA and RNA. Each of them follow the same set. Even viruses, the ATGCs of your DNA code are read by ribosomes, and if it says UUU, that is the only amino acid that gets formed in every molecule. So there's something very constant here, but then we don't know for sure if this is the same way life would be elsewhere, because our life is organic and carbon based. But if we go to some other planet, will that be exactly the same? And how many of you didn't watch Project Hail Mary? Oops, okay, then I should not spoil the movie for you. But yeah, we know the movie, Ryan Gosling, Grace, goes to space and he befriends a rock, an alien rock called Rocky. And the life form there is not carbon based like ours, it's ammonia based. So there is a question, if life really started elsewhere, would it necessarily have to start the same way as ours, or can it form in other ways too? The movie made a decision by saying it's not, and we can find rocks also alive. But unless we really go there and find, we don't. And if anyone in the future is ever given the chance to get to go to space and you don't, please enroll me. I would like to take part in that, and I'll make the sacrifice if I can go meet Rocky.
Will you be able to handle that?
Sure. So okay, wrapping up, we zoomed out into the universe, and then we zoomed in. We came out to see other planets where life could exist. But then now we'll get back to understanding. We have a better understanding now of how everything works. So let's get back to our important question: why are you alive? I've been using this a lot as a selling point to call you guys here, and thanks a lot for that. So we've all seen this image. It's the pillars of creation, and on the left it's a different wavelength, and on the right it's on the infrared range. You can see them differently. We've evolved to we have a certain wavelength to see it, so we see it differently. But then other life won't see it the same way. But it was so all the atoms we got ordered in a certain way, and if we don't have this certain order, we would have just been stardust, which is atoms in space. But what I want to say is we are alive because we are the universe trying to understand itself. Following the second law of thermodynamics, what the universe basically did is make something that could self-replicate and grow and become complex enough that it could get curious to wonder about itself. And maybe this wonder is helping it increases entropy more. I don't care about that, but it's really fun to try to understand the universe. This line has so many layers in it, because it says that we are the universe itself and we are trying to understand itself. And you guys have come here and all these years matter just existed. It was there, stars and atoms, it was all crashing, huge events were happening, but not no one was there to care. But now we are there to see and care, because we got complicated enough for the universe to look at itself. I don't know if this is selling enough for you guys, but it should be. So this is also a very interesting line. RNA didn't just survive, it built eyes to look at the universe. It has limitations. We see a very limited spectrum because of the shortcomings of the planet we are in. But I wish in the future we spot life, or we could turn into life that could behave as a spectrophotometer, which can see in any wavelength, or behave as an acoustic, as a microphone which can detect any frequency. And if we really turn into that, would we really be ultimate beings? So yeah, we are the universe trying to understand itself, and I think the fact that all of you guys took your time to come this evening and ponder upon this question really explains the fact that my answer is right. You guys are really trying to understand the universe, and that's why we are all here, guys. So that's what evolution has really done this for. Thank you. Thanks a lot. I understand my talk was very, very, very unconventional. I went on random tangents. I went on tried to connect everything, because I wanted to pay tribute to my OGs, Vsauce and Veritasium music. And I wanted to frame a top, the title of the talk was something like a Vsauce video, One of the previous hypothesized title was the molecule that shouldn't exist but still exists. So that which is RNA. But still, why are you alive was a good enough title to pull you people. And yeah, the whole idea was to connect these different topics and show that there's a link and you can answer the big question by stitching them all together. And yeah, I really have to thank you all. Like, okay, an applause for you guys because thank you for coming here and listening to my talk. Yeah. I thank Horizon 23 batch and 24 batch because I don't know which batch tried to organize this session. This is a 10-year shift, guys. That is why it's so. So yeah, I'll end it here. Anything you want to talk to me about all this? I also just figured things out. We can have a conversation. Yeah, bye.